Welcome to my channel and in this video we will be talking about sexual reproduction in bacteria. See, bacteria exhibit a very primitive form of sexual reproduction, that is there is no gamete formation or fusion, but one of the essential feature of the sexual reproduction, that is genetic recombination, does takes place in this. So there are three methods by which the genetic recombination is achieved, and these are transformation, conjugation and transduction. So let's start with transformation. This experiment was done by Griffith in Diplococcus pneumoniae bacteria. Keep this in your mind that in transformation, donor and recipient do not come in contact. Okay, the donor cell releases a piece of DNA which is taken up by the recipient cell from the solution, and this ability to pick up the DNA is called competence. See, there are two strands of this bacteria, S3 which is virulent one and R2 which is non-virulent one. So four steps were performed in this experiment. Number one, when S3 which was virulent bacteria was injected into healthy mice, mice died. When R2 which was non-virulent one was injected into healthy mice, mice survived. But when the heat killed bacteria which lost its virulent capacity on heating was transferred into this healthy mice, mice survived. But when the combination of R2 and the heat killed S3 bacteria was injected into this healthy mice, then mice died. So why this happened? So by this, Griffith concluded that there was something that passed from the heat killed S3 bacteria to R2 bacteria, so that the non-virulent one was converted into virulent one. So later on, three scientists, Avery, McLeod and McCarthy, repeated this experiment using various enzymes, and they proved that the transformation principle was DNA. And they also proved that DNA is the genetic material. So let's start with conjugation. This experiment was done by Lederberg and Tatum in E. coli bacteria. See, there is a special type of plasmid found in this bacteria called the F plasmid. Here F stands for fertility. The bacteria which has this F plasmid is called donor, F plus or male cell, and one which lack this plasmid is called F minus cell, recipient or female cell. This female cell may contain any other plasmid, but they lack the F plasmid, that is fertility plasmid. The bacteria which has this F plasmid, that is F plus cell, produces a proteusant called the sex pili, and with the help of this F plus cell attached to the recipient cell and transfer its replicon of plasmid to the recipient cell through conjugation tube and converting it into the F plus cell. So this is how the transfer of genetic material takes place. So sometimes what happens that due to defect in this conjugation tube, there only a part of plasmid get transferred into the other cell and get incorporated into plasmid cells, and that cell is called HFR, that is high frequency cell or supermel. So this cell which is having the foreign material will always wanted to get rid of this foreign plasmid. So there is always increased efficiency of genetic transfer. So whenever we use this HFR cell as a donor, the frequency of genetic transfer increases. Like when conjugation takes place between F minus cell with supermel, then frequency of recombination increases by 1000 times. Okay, so the next third process is transduction, and we will see this in the next video. Thank you.